Right, okay, so number 4 in the series of short lectures, come videos on some of the concepts you did last year in biochemistry, just to remind you, because many of these concepts are important to underlie what we're doing in food science and chemistry. Okay, reactions. Here's a schematic reaction of a schematic of a biological reaction. During a biochemical reaction, substrates, sometimes called reactants, are converted into products. The conditions of reaction are important. Most biological reactions take place under physiological conditions. So, for example, pH 7, whatever the body temperature is, if it's an animal, the ambient temperature, often if it's a plant. Often other substances, such as ATP, are needed, and almost all biological reactions require enzymes. Most biological reactions are reversible, as indicated by the 2 half arrows. Most biological reactions take place in the context of metabolic pathways, that is to say, sequences of linked reactions. The low diagram there is a little bit of the first part of glycolysis, which converts the 6-carbon glucose molecule into 2,3-carbon pyruvate molecules. And there's also a reverse process called gluconeogenesis, which does the opposite. In the schematic we've got at the top, we've got A and B being reactant to reversibly form C, then reversibly form D, and reversibly form E. The rate of these reactions depends on conditions. Often, if we're short of E, the reaction will go in the forward direction. If we have a lot of E, bodily systems, the reaction will go in the reverse direction. Again, more of this in human metabolism. So, some important reaction classes. Synthesis and hydrolysis underlie life. Synthesis is how we make big molecules. Hydrolysis is how we take big molecules and make small molecules out of them. When we eat food which contains many large and complex biological molecules, we need to break these large molecules down, that is to say, hydrolyze them, to produce small molecules which may then be used to synthesize large molecules, or they may be used for things like energy, for example. We'll look at this in specific detail when we look at proteins and starch a bit later on in the module. This is arguably the most important hydrolysis. Without this, we wouldn't be doing anything. We eat and we breathe to regenerate the molecule ATP. When it's hydrolyzed to produce ADP and phosphate, it liberates energy, which allows other reactions to take place. And as I said, this is literally why we eat, and this is literally why we breathe, to do this hydrolysis reaction. Okay, monomers and polymers. Biological macromolecules, sometimes called polymers, are made up of simple repeating units called monomers. You'll sometimes see in the textbook the phrase like an amino acid residue. What that means there is that an amino acid has been incorporated into a protein molecule and is henceforth referred to as a residue until it's hydrolyzed out, perhaps. So the first example here, where all the monomers are black, these are identical monomers forming a polymer. So, for example, this may be glucose forming a small part of either cellulose or starch. In the second example, we have different monomers forming a polymer. An example of that would be amino acids forming a protein. Okay, oxidations. A number of different ways to describe oxidation and reduction, the simplest of which is addition to oxygen. So this is happening here. If you look at the carbon atom in C2H6, when it starts in the reaction, it has hydrogens attached to it. When it completes the reaction, it has oxygens attached to it. So the carbon has been oxidized. Meanwhile, the oxygen molecule here has been converted to water, so it's said to have been reduced. Now this is the classic redox cycle. All oxidation reactions are associated with a reduction reaction, what we call a redox process. Again, as I'm sure you'll do in biochemistry, in human metabolism, what we call oxidative metabolism, converting carbon atoms into CO2 using oxygen, is a reduction oxidation process. Quite a complicated one, but there it is. Okay, so that's one definition of oxidation and reduction. Oxidation is a gain of oxygen, in this case to these carbon atoms, or alternatively a loss of oxygen. On the other hand, it can also be, again, oxidation can also be defined as a loss of hydrogen whereas reduction is a gain of hydrogen. So in this case we've got ethanol, which has been reduced, which has been oxidized rather, in the two-step process which takes place in the liver by two enzymes. In this case the oxidation is similar to the previous slide, but it's accompanied by the reduction of the molecule NAD to NADH. So if you look at the carbon atoms, this carbon atom here, for example, at the moment has got two hydrogens attached to it. Gets converted to this molecule, acetaldehyde, which now has one hydrogen attached to it. I beg your pardon, one hydrogen attached to it, so it's lost a hydrogen, so that's an oxidation. The second stage, using aldehyde dehydrogenase, attaches another oxygen, so that's another oxidation that's taken place. On the other hand, this big molecule NAD, which I haven't drawn the whole thing of, because as I'm sure you know, it's quite a big molecule. In this case, we're adding a hydrogen to it, so that's a reduction. We do this twice, and that again is a reduction. Okay, so oxidation and the reduction. Oops. Okay, enzymes, just a little bit of revision on enzymes. I'm sure you're familiar with this, just what they do. Most biological reactions are very slow indeed if left to themselves. Enzymes, which are mostly molecules that greatly increase the rate of reactions, that is to say catalyse them, without themselves being changed at the end of the reaction. Biological molecules are three-dimensional, and enzymes will only work with the molecule they have evolved to catalyse, called stereospecificity. So we've got at the moment we've got there a molecule which doesn't fit, trying to latch onto the enzyme, and it can't, so no reaction takes place. It'll cycle around in a second. There's the enzyme back again with its two active sites, which in this case its substrate will bind to. A reaction takes place, in this case a hydrolysis, and the two smaller parts of the molecule after the hydrolysis are released. Okay, so that's a hydrolysis. Here's a animation of a synthesis taking place. Two substrates come together, a reaction takes place, and then the new larger molecule is released, leaving in both cases the enzyme in the original condition it was in. Okay, most all enzymes are globular proteins, as I'm sure you know. There's some RNA enzymes. Quite big molecules, very much bigger than their substrate normally. Definitely require a cofactor, often, in fact, almost always a metal. We'll discuss this when we talk about the enzyme polyphenol oxidase, which has copper as a cofactor. So the active site. This is an example of a enzyme which performs it's a proteolytic peptidase which breaks peptide bonds of non-terminal amino acids, i.e. within the molecule. And it has right at the centre, it has a metal as a metal at the centre, which is zinc in this case. And there, the green there is the biological molecule binding to it, ready to be, in this case, hydrolyzed. So to summarise, an enzyme binds to its substrate to form an enzyme-substrate complex. This binding is reversible, as indicated by the two half arrows. The enzyme then releases the product, and the enzyme, again, in an irreversible step. We refer to these as substrate binding in the first step, and the catalytic step in the next one. Okay, that's all for that. Hope that was useful.